This video will be the first video on transcription. So what I'll talk about is the types of RNA and the structure of ribonucleotides and also the structure that RNA forms. Then I'll talk about the coding strand versus the template strand, and we'll talk about methods for assessing whether a particular gene or a group of genes is transcribed into RNA. So we now move into the central dogma. And so the central dogma is that information flows from DNA to RNA to protein in cells. And transcription is that process by which the information is taken from the DNA and copied into RNA. There are a couple of points I want to make about how this is different in prokaryotes versus eukaryotes. So prokaryotes, remember that it does not have a separate nucleus. And the result of that is that the RNA, which is shown in red here, is just freely present in the cell, and as soon as messenger RNA starts to be made and starts to be synthesized, it immediately gets bound by ribosomes and starts being translated. So everything kind of happens in the same compartment here, and it happens simultaneously as well. In contrast, in eukaryotes, transcription happens in a separate compartment, the nucleus, and RNA is made and then is processed before it is exported out of the nucleus. And so the RNA is really completed before it goes out and gets bound by ribosomes for translation. So there are a number of different types of RNAs, and you'll see a table here that looks very complicated about many types of RNAs. What I'd like to highlight is that there are really two main types of RNAs that we will consider. So the first type is messenger RNAs. And so messenger RNAs are the ones that code for proteins. So they are transcribed from the DNA, they go out into the cytoplasm, and they are bound by ribosomes, and they encode a protein. The other RNAs all fall into this class that we call functional RNAs. And in this case, the RNA is in fact encoded in the genome. So it is transcribed from the DNA into RNA. However, that RNA does not code for a protein, and the final product of that gene that is being transcribed is actually the RNA. And so RNA itself has many essential roles in the cell. Some of them I'm sure you're very aware of, for example, transfer RNAs, ribosomal RNAs, but there are a lot of other varieties of RNAs as well that fall into this class of functional RNA. So RNA is made of ribonucleotides, and oftentimes people get confused about the differences between deoxyribonucleotides and ribonucleotides. So the nucleotides that make up RNA are different from the nucleotides that make up DNA in two essential ways. The first way is that the sugar is actually a different sugar. It is very, very similar, but it is not the same. So it is a ribose sugar, and I'll highlight the difference between that and the deoxyribose sugar on the next slide. And so all of the ribonucleotides do in fact use that ribose sugar. The other difference is with one of the bases. So you'll notice that adenine, cytosine, and guanine are the same between DNA and RNA nucleotides, but RNA nucleotides use the base uracil. Uracil is very, very similar in structure to thymine, and uracil is used instead of thymine in the RNA. The only difference is that it's actually missing one methyl group that is found in thymine. Overall, however, I just want to highlight that ribonucleotides are made up of the same three components. They have the sugar, the ribose sugar, the phosphate group, and the base. And the relative structure is pretty similar. For example, the base attaches to the 1' carbon of the sugar, and the phosphate attaches to the 5' carbon. So this highlights the difference between ribose sugar and deoxyribose sugar. Please note that they're very similar, but the one difference is that on the 2' carbon of ribose, there is an OH instead of just a hydrogen that is found on deoxyribose. This very small difference in the sugar pretty profoundly affects the molecules that are made from ribonucleotides. And so RNAs, instead of being very stable like DNA, is relatively unstable. Furthermore, ribose prefers to be single-stranded instead of double-stranded. So ribonucleotides hook together also in the very same way that deoxyribonucleotides hook together to make DNA strands. So you see here's one ribonucleotide base right here. There's the sugar, there's the base, and here's the phosphate. And new RNA nucleotides are added onto the 3' carbon of that nucleotide. And so here is a strand. And if you were going to add a new nucleotide, it would get attached onto that 3' carbon via that 3' hydroxyl group. Also, this is just an example of the RNA structure that can form. So we'll generally write RNA starting at the 5' end and going to 3'. It is single-stranded. However, it does in fact often fold itself into partially double-stranded structures. And so there's some hydrogen bonding that can take place within that molecule. That bonding follows the same rules as is found in DNA, so C's with G's and U's with A's. However, it does not form into an entirely double-stranded structure. So when we have DNA is double-stranded, and the information to make RNA comes from the double-stranded DNA. This right here it's called a transcription bubble. So DNA will open up and you have 2 strands of DNA. And really importantly, for a particular gene, only one of those strands is ever used as the template to make a particular RNA. If you look at these 2 strands of DNA, I just want to point out that the complementary strand of DNA would be this particular sequence. I just wrote the complementary strand for this AGC, etc. strand down here. And I want to highlight that any RNA that was made by using this bottom strand as a template would actually have a completely different sequence. It would have a different RNA sequence, and that RNA sequence would code for completely different amino acids if it was a protein coding gene. And so if you're making a particular gene, it's really only one of the strands that is ever used to make that particular RNA. These strands have names. So here is double-stranded DNA. If you look, you see that the bottom one is complementary to the top strand. One of those strands is always used to make the RNA. And so in this case, it is actually the bottom strand that is the template strand. So the template is the one that is used to make the RNA. And you can see that the RNA is complementary to the template strand. So if you have a G, a C is called for. If you have an A U is called for. Now, what I want to highlight is that the template strand is really the strand of DNA that is essential for making RNA. However, when we look at the DNA sequence, what you'll notice is that RNA sequence looks very much like the other strand, right? So even though the template strand was used to make that RNA, if you look at the sequence of the RNA, it really looks identical to the other strand of the DNA, except that there are U's instead of T's. And so the other strand that actually looks like what the RNA is going to look like, we give it a special name as well. Sometimes it's called the non-template strand, but more frequently we call it the coding strand. And we call it the coding strand because it tells us what that RNA is going to look like. Now, if you look at entire chromosomes, you will find multiple genes arrayed along that chromosome. One important thing is that it is not always the same strand of the DNA along a chromosome that is the template strand. So for a particular gene, one particular strand is always used as the template strand, but a different gene might use the other strand as a template strand. So genes can use one or the other, but they always use the same one for that particular gene. One other thing I'd like to point out is that the strand that is used determines which direction transcription is going to proceed in. So if you look at this first gene, gene A here, the bottom strand is the template strand, and the beginning of the gene is found right here on the left side. RNA is synthesized in an anti-parallel manner, and RNA always starts at the 5' end and adds new bases onto the 3' end. So therefore, while RNA is synthesized in a 5' to 3' direction, the DNA template strand is actually read by the transcription machinery in a 3' to 5' direction. Gene B uses the top strand as the template. It is read in a 3' to 5' direction, and so therefore we're going to start synthesizing the RNA over here on the right, and we will proceed toward the left side. So one type of problem I might give you to check if you understand these ideas of the directionality of transcription and the idea of the template versus the coding strand might be a problem like this. So when the following double-stranded DNA sequence is transcribed into RNA, what is the RNA sequence? So here you have to remember a few things. First, the template strand is the strand that is used to make the RNA, and the template strand is read starting at the 3' end and going toward the 5' end, so that the RNA is produced starting at the 5' end and going toward the 3' end. And so that means that the strand that would be used, so this top strand is the template, and that C will be the very first base that is transcribed into RNA. And so opposite of a C would be a G. This would be the 5' end of the RNA. And the next one would be another G. And then opposite of an A would be a U, followed by another U, an A, and a C. The last topic I'll talk about in this video is the question of how we investigate whether particular genes are being transcribed. So this is a really important question. As you all hopefully know, every single cell in our body has the very same genome. So all of the genes are identical, whether a cell is a muscle cell or a neuron. But those two different types of cells have extremely different functions that they need to carry out. And the basis of the difference between the different types of cells in our body with vastly, vastly different functions is really which genes are being used within those cells. And so which genes are being transcribed into RNAs and translated into proteins. So we'd like to talk about how do we assess whether a particular gene or genes are being transcribed into RNA or being expressed. I'm going to tell you two experimental approaches to look at an individual gene, a particular gene that you have in mind. Then I'll tell you several experimental approaches that are used to look at many, many genes simultaneously. So the first method I'll talk about is a northern blot. So we already discussed what a southern blot is, and it is used to determine whether DNA with a particular sequence is present in your sample. And if you recall, we took the DNA, we ran it on a gel, and we used a radioactive probe that was complementary to the sequence we were looking for. So a northern blot is very, very similar to a southern blot. It's just using RNA instead of DNA. So the way this technique is done is that you take cells and you extract the RNA from them, and then you separate the RNA on gel electrophoresis. You separate it by size. Then you would transfer that RNA onto a membrane, and you would use a radioactive probe that was complementary to the messenger RNA that you were looking for to determine whether that particular sequence was present. And the way this looks is something like this. So oftentimes people would take RNA from many different tissues, for example, all of these different tissues across the top, and then they would investigate one or a couple of different genes. And so here, this particular gene, PSAT, you would see a band if that messenger RNA was present in that particular tissue. And so you can see that that particular gene is very high, for example, in the pancreas and the kidney, but appears to be absent in skeletal muscle, lung, placenta. A much more commonly used technique these days is something called RT-PCR. And so we talked about polymerase chain reaction already, and that is just a way to amplify a given piece of DNA to make lots of copies of it. So RT-PCR really includes one extra step before you do the PCR, and that is a reverse transcription step. And so RNA is isolated, and a enzyme called reverse transcriptase is used to make a DNA copy of that RNA. Once you have the DNA, then you can amplify it by PCR, and you use primers that are specific to a particular gene. And so generally RT-PCR will give a present or absent result. So this is an example of what that might look like. This particular gene, NANOG, has been investigated in these different tissues, and you can see that it is present in the testis and the ovary, but it is absent. There's no band at all in any of these other tissues. The last two techniques I'll talk about are a way to investigate not just one or a few genes, but really all of the genes that are being transcribed in the entire cell. So you've probably heard the term genome, and so the term genome is really used to indicate all of the genetic information that's present in a particular species, so all of the genes that are there. There's another term that is commonly used these days. It's called transcriptome. And a transcriptome is specific for a particular cell or population of cells. And it is simply all of the genes that are actively being transcribed in that certain population of cells. So there are two ways that we can investigate the entire transcriptome. There's one way called a microarray, and this is also known as a gene chip. A gene chip would have something like 1,000,000 different spots on it, and each spot has a little cluster of DNA molecules that have a particular sequence that is the same as one of the genes in the genome. The way this works is that this gene chip has this grid of lots of little spots of DNA, and you start out with RNA from your tissue of interest. You would add a fluorescent molecule onto that RNA, shown over here, and then you would hybridize that RNA to the gene chip. That fluorescent RNA can then bind to particular spots on that gene chip, and wherever it binds, it brings in a fluorescent signal. So therefore, certain genes that have very high levels of messenger RNA in your sample will have a bright fluorescent signal, whereas other genes that are not being transcribed at high levels in your particular sample will appear dark. This is actually what the array looks like. It's microscopic, and it is read by software that detects the fluorescent signal. Finally, the last way that we can look at the entire transcriptome is by taking all of the RNA that is found in a cell and simply reverse transcribing that RNA and sequencing it. Turns out that sequencing has become so very cheap these days that this is actually the method of choice these days, just to go ahead and sequence all the messenger RNAs that are found in the cell in order to determine which genes are being expressed.